Please welcome Taiwo Ojo from Nigeria, and he's going to talk about repurposing FDA-approved cardiovascular drug to inhibit rage in Alzheimer's disease through virtual screening and molecular dynamics investigation. Thank you so much. Welcome to BSI, and please go ahead and present your work.
All right, Sarah, thank you. My name is Ojo Taiwo Ojo. I'm from Nigeria, and I will be presenting on one arrow in our arsenal proposing FDA-approved drug for the treatment of Alzheimer. Now, I would like to go ahead to share my screen now.
Perfect.
Okay, so like I said, my name is Ojo Taiwo once again, and I'm a research assistant from the Computational Molecular Biology and Drug Discovery Laboratory, Department of Biochemistry, Ladoke Akintola University of Technology, Ogbomoso, Oyo State, here in Nigeria. And I will be presenting on one arrow in our arsenal. We're proposing FDA-approved cardiovascular drugs to inhibit rage in Alzheimer's disease through virtual screening and molecular dynamics investigation. Now, to begin with, I would like to give introduction to what Alzheimer's disease and rage is in particular. As we all know, Alzheimer's disease is the most prevalent central nervous system amongst many other that we have, the likes of schizophrenia, multiple sclerosis, neuroinflammation, and some other CNS-associated disorders. But among all of them, we have Alzheimer's disease to be the most prevalent. And one of the notable biomarker for AD is the plaguing of the insoluble beta amyloid. Now, this beta amyloid can come in many variants as research has widely investigated them. We can have the beta amyloid in 40 amino acid residue peptide. We can have the one of 42 amino acid residue peptide, and we can have the one of 42 amino acid residue peptide with a phosphorylated serine at position number 8. And also the fourth one, we can have an isomerized aspartate at position number 7 on the 42 amino acid peptide of beta amyloid. Now, what is the role of rage in Alzheimer's disease when it comes to Alzheimer's disease and rage? Now, rage is an acronym that stands for the receptor for advanced glycation end products. And this rage is a multiligand protein which contains up to like 3 domains. We have the V domain, the C1 domain, and the C2 domain. Now, it now depends on the ligand that binds to each of these domains to elicit a pathological condition. For example, if beta amyloid binds to the VC1 domain, it results into Alzheimer's disease. If HMB-GB1 protein or S100 protein binds to either the V or the C1 domain, it can also lead to neuroinflammation, and also the advanced glycation end product, AGE. If they bind to the V domain of rage, it can also lead to diabetic neuropathy, nephropathy, and some other pathological conditions. So just in summary, rage is a multiligand protein, and then it depends on the kind of ligand that is binding to it to elicit a pathological condition. Now, what is now the role of rage in Alzheimer's disease? When rage found in the extracellular matrix, when it binds to beta amyloid, it transports beta amyloid into the microglial cells of the brain, and then it's allowed beta amyloid to aggregate and form a plaque around the synaptic cleft. This disrupts transmission, neurotransmission. It disrupts the passage of information from one neuron to another neuron. And then this disruption can be marked by deterioration of brain cells, cognitive decline, progressive memory loss, which can lead to visuospatial disability or even cognitive disability. Now, what is the research gap? Now, as we know, scientists are trying all efforts to mitigate this menace caused by rage and beta amyloid, but all of the efforts put in place ever since rage has been noted to be one of the culprits in Alzheimer's disease. All the efforts have been futile. It has always been in vain. And one of the recent unsuccessful efforts is the failure of accelerator guns in 2018. Azelagragon was proposed to be a RAGE inhibitor to stop Alzheimer's disease. But when it got to the second phase of clinical trial, this drug actually failed to perform up to its optimum therapeutic efficiency. So up to date, there hasn't been any successful inhibitor of RAGE to stop the menace posed on Alzheimer's disease. Now, the research methodology. This is a computational investigation that investigates up to 230 FDA-approved cardiovascular drugs. Now, this research collated all the 230 FDA-approved cardiovascular and then screened them against the VC1 domain of RAGE. This is a region. The VC1 region is the region where the beta amyloid peptide binds to and where it got transported into the CNS. Now, the screening of these cardiovascular drugs leads to identification of a particular approved drug, pravastatin, to have a good inhibitory prowess on the RAGE VC1 domain. Now, the addition of this particular compound led to its optimization. This compound was further optimized to increase its therapeutic efficiency. And then after its optimization, the optimized version of pravastatin was also used to generate more random molecules, up to like 10,000 of them, to create a library of compound. Now, among these 10,000 molecules, a lot of filtering parameters was done on them, like the Lipinski rule of 5, their pins ability, and some other filtering parameters. They were subjected to these filtering parameters, and then in all of these 10,000 compounds, only about 238 was able to scale through this filtering parameter and then subjected to the second phase of molecular docking against the RAGE VC1 domain. Now, while experimenting their binding affinity and then their interaction prowess on RAGE VC1 domain, only 3 compounds stood out among these 238 compounds. And then these compounds are compound 67, compound 183, and compound 213. And in all of these, the 3 compounds, they were also then subjected to pharmacodynamics and pharmacokinetics investigation. That is their ADMET property. Then also they were investigated through molecular mechanisms with generalized bond surface, MMGPSA, and then up to the last stage of molecular dynamics to test their stability and then how tightly fitted they are in the pocket of RAGE VC1 domain. Now, of the results gotten during the investigation, like I said while introducing, beta amyloid can come in up to like 4 variants of them. We have the beta amyloid of 40 peptide amino acid residue. We have the one of 42 amino acid residue. Then we have the one of phosphorylated serine. And then we have the one of isomerized aspartate. Now, to check whether that is where the title of the research came from, one arrow in our arsenal, to check whether a single compound or a three compound can be able to tightly prevent all these 4 variants of beta amyloid from binding to RAGE VC1 domain. A molecular docking investigation was done between all the variants of beta amyloid and RAGE VC1 domain to check whether all of them have the same binding pose and binding interaction with RAGE VC1 domain. And fortunately enough, all of these beta amyloid variants have the same interaction profile against the RAGE VC1 domain, which implies that if we can identify a single compound to inhibit that particular pocket, it will prevent all the RAGE or the beta amyloid variants from binding to RAGE VC1 domain. And so this is their binding affinity score, making beta amyloid with amino acid residue to rank the table, and then the one of isomerized aspartate to be below the table. The amino affinity was not really that important than the interaction profile they exhibited, but the interaction profile they exhibited showed that they seemingly have the same interaction interactive residues against RAGE VC1 domain. And of note is the one particular amino acid residue that contributed to the interaction, which is lysine 37. We found out that lysine 37 was very important in this interaction against RAGE VC1 domain. Now, identification of best hits, which is pravastatin. Pravastatin, during the molecular docking of the 230 FDA drugs, pravastatin stood out in the molecular docking, and then it was subjected to two types of molecular docking procedure: the SP, which is the standard precision, and then the XP, which is the extra precision. The extra precision docking was done on pravastatin just to make sure that it's not giving a false positive result. And as we have it, the XP docking actually revealed more the therapeutic efficiency or the inhibitory power of pravastatin against the RAGE VC1 domain. And one of the interaction that we are much more interested in is the lysine 37, as we can see here. So the XP docking showed that pravastatin can form a salt bridge and also an hydrogen bond with lysine 37 on the RAGE VC1 domain. Now, optimization of pravastatin. During the molecular docking, we were able to get that some compounds were able to form a salt bridge with glutamate 32, which pravastatin did not form. And we were able to know that one of the functional groups that form this salt bridge is NH3+. So NH3+, pravastatin was optimized on its carbon 1 to form to have this functional group NH3+ to see and investigate if it will have the same salt bridge other compounds we're having with glutamate 32, which is also one of the responsible amino acid residues stabilizing the molecular interaction between RAGE and beta amyloid. So the pravastatin was further optimized, and then pharmacophore modeling was performed on pravastatin to generate up to 10,000 compounds. And then of the 10,000 compounds, after filtering, we're able to see 238 to outstand the rest of them. And then this 238 was subjected to molecular docking investigation against RAGE VC1 domain. And fortunately, the binding affinity of all of these optimized pravastatin pharmacophore actually have better binding affinity and even more interaction prowess, more than pravastatin. So it's like we have here, pravastatin has minus 4.8 kilocalorie per mole binding affinity, while its pharmacophore actually generated minus 6.5 for compound 67, minus 6.1 for compound 183, and minus 6.0 for compound 211. And then we can see that all the hydrogen bond interaction and salt bridge together with hydrophobic interaction were also very much retained in the interaction, except compound 183, which does not have lysine 37 interaction. The remaining two compounds actually have lysine 37, and they form a salt bridge and also an hydrogen bond. But like compound 183 also forms a salt bridge with lysine 37, except the hydrogen bond that it didn't form with it. So it shows that this pharmacophore of pravastatin have more prowess than pravastatin itself, and then they can be a better inhibitor of RAGE VC1 domain to mitigate beta amyloid from binding to RAGE. Now, their ADMETs and toxicity results also show that these compounds are of therapeutic, they can be of therapeutic purpose because the absorption parameter were okay, their distribution parameter too were very much okay, and then they are under their metabolism. One of the important isoform of P450 cytochrome P450 enzyme is cytochrome 34A. So we could see that none of these compounds have the ability to inhibit this particular enzyme, and then their metabolism is quite all right in this regards. They will serve as a good therapeutic or druggable compound against RAGE in Alzheimer's disease. Their excretion, which is the human clearance too, also show better values, because whenever we have a positive value, that means their clearance rate is low, but whenever we have a negative value, rather, pardon me, so it shows that their clearance rate is low. And then their toxicity too, except for the hepatic toxicity and then DILI, which was also observed in pravastatin, because they are pharmacophores from pravastatin. All other toxicity parameters were very much okay. Now, the free energy calculation, this was just to investigate further the binding affinity and then how tightly this compound will be in the pocket of RAGE VC1 domain. And as we have it, the compounds really performed the same way they performed during molecular docking, with compound 183 coming as the best ranked compound here with minus 45.01 delta G value. And also we see their solvation energy, their van der Waals forces, and then the contribution of hydrogen bonds to the binding affinity. Now, during the molecular dynamics simulation, we're able to investigate this compound to the apoprotein, the unbound state of the protein. We compared it to the bound state of the protein. The black spectrum represents the bond state of the protein with compound 183, while the red spectrum represents the bond state of the protein with compound 211. The green spectrum represents the bond state of the protein with compound 67, while the blue spectrum represents the unbound state of the protein, just the apoprotein alone. So we can say that compound 67 and the apoprotein likely their graph spectrum was almost the same. That means the compound, the bond state of the protein and then its unbound state also have the same spectra. They have the same RMSD value. But we can see that compound 211 is likely to gyrate or likely to be destabilized at the 40th nanoseconds up to the around 80th nanoseconds during the process of the simulation. But the RMS of fluctuation, root mean square of fluctuation, show that all the bond states and the unbound state of the protein likely have the same fluctuation spectra, because there was no noticeable spike in the spectrum. So this is depicting that whether the protein is bound or the protein is in its unbound state, the protein will the binding of the compound will unlikely make the protein to fluctuate. It further stabilizes the protein even in its bound state. Then the hydrogen bond contribution. These are the contribution of hydrogen bonds for each compound from compound 183, 211, and compound 67, which makes the green part is much more prominent in the graph, showing that compound 67 have the highest hydrogen bond contribution during the process of simulation. And then the radius of gyration also, compound 67 and compound 183 have a very low radius of gyration, even compared to the unbound state of the protein. This is showing that the protein further stabilizes, the compound further stabilizes the protein, and then the compounds being really gyrate out of the pocket of the protein, which shows that even when there is a competitive force from beta amyloid, this protein and the compounds will still likely have to be tightly bonded together even in the presence of beta amyloid. So whether we have a competitive binder or we don't have a competitive binder, these two compounds will likely have to retain their binding affinity prowess to the pocket of RAGE. In conclusion, the virtual screening was able to pinpoint 3 pharmacos of pravastatin, which were taken through the molecular docking, ADMET, RO5, molecular mechanics, and molecular dynamics simulation. So the evaluation of these compounds showed that they can be RAGE putative binders, and they can be a druggable compound in inhibiting the binding of beta amyloid to the pocket of RAGE. I would like to acknowledge and appreciate Beyond Science Initiative for giving me this opportunity to present this year. Much thanks to the coordinator and all the BSI team. Also, I would like to appreciate the ComChem Hub, where I also am director of training at that institute, and also where I'm serving as a research associate, the Computational Biology and Drug Discovery Laboratory, Department of Biochemistry, Adeoke University of Technology. I would like to say thanks to all the team member and my colleagues. Thanks for listening.